How's that for sound? Can you guys hear me okay? Does that sound good? All right. Okay, well, we have a fun session for you, so I'm going to go ahead and get started with an introduction so that we can get the most out of our speaking time. Thank you for coming to our session, Wild Kingdom, the Scope and Diversity of Animal Phenotyping-Related Core Facilities. My name is Ingrid Bergin. I'm the session chair for today. I'm a veterinary pathologist and a professor in the Unit for Lab Animal Medicine at the University of Michigan in Ann Arbor. And I'm also the director of the ULAM Pathology Core, which provides histopathology services, including, I would say, morphology-based phenotyping of animal models. So the impetus behind this session was to expand the visibility of animal phenotyping-related cores within the umbrella of ABRF. And so one broad definition of phenotyping that's commonly applied would be the characterization of observable traits that result from the interaction of an organism's genes and the environment. So that seems pretty broad, and I think you can understand that within the context of that definition, phenotyping can actually mean some pretty different things to different people and in different contexts. So even though this is, I think, the only session that has a specific reference to animal phenotyping in the title, if you think about the topics and some of the talks that you've seen so far and that are on the agenda for this meeting, there are actually a lot of sessions that deal with animal model phenotyping in one way, shape, or another. But most of them are doing so in the context of answering a specific question or from a specific subfield, like be it spatial biology or genomics, or morphology-based, as in my case. So I think the advantage of having an animal phenotyping focused session is to encourage us to consider animal models in a more holistic way and to give us some resources for accessing large databases or other types of phenotyping that might be a useful complement to how we approach animal model characterization in our own specific subfields. So that's part of the intent. And to that end, we have two highly expert speakers today. Dr. Lloyd from the University of California at Davis, who will be speaking about large-scale mouse phenotyping consortia, and Dr. Eugene Chen from the University of Michigan in Ann Arbor, who will be speaking about genetically engineered rabbits as an example of a non-murine animal model in translational research. So the hope is that these sessions will provide useful information whether you're currently working in an animal-focused core or if you're working in another context but utilizing animal models. And the hope is that you'll get some better understanding of animal model phenotyping protocols and resources from this session. And then finally, my hope is that this will kickstart future animal model-focused cross-disciplinary discussions and collaborations amongst ABRF members. So, a couple housekeeping things. The structure of the session is we have two speakers, as I mentioned. Both talks are about 30 minutes or so. Depending on how we're doing for time, we may pause between for a question or two, but I'm hoping there'll be 10 minutes or so left at the end, so we can do kind of more broad-ranging questions at the end. Both speakers are utilizing either patient-based material or some unpublished material, so I do ask that you refrain from photographs of the screen, but both of our speakers have agreed if you have questions afterwards or if there's specific slides that you would like access to, you can feel free to contact them and they can provide you with information that's free to share, okay? So, our first speaker is Dr. Lloyd of the University of California at Davis. So, as a fellow veterinarian turned comparative biologist, I'm proud to say that Dr. Lloyd was originally trained as a veterinarian and received his DVM from the School of Veterinary Medicine at UC Davis. He then earned a PhD in physiology at the School of Medicine at UCLA, and he has been on the faculty at UC Davis for 28 years, first in the School of Veterinary Medicine, and then in the School of Medicine, where he is now a distinguished professor in the Department of Surgery. He is also the Associate Director of Shared Resources of the UC Davis Comprehensive Cancer Center and Director of the UC Davis Mouse Biology Program. He is an internationally noted expert in mouse biology and genetics who has expertise in the production and phenotyping of specific gene-targeted mouse models for the discovery of disease-specific biomarkers and translationally relevant phenotypic investigations. Dr. Lloyd is the principal investigator and the project director of the Knockout Mouse Project, or COMP2, and the Mouse Metabolic Phenotyping Center, or MMPC Live, at UC Davis. And today, he's going to provide us an overview of mouse phenotyping services at UC Davis, specifically in the context of large collaborative phenotyping projects like COMP2 and MMPC Live. So I'd like to welcome Dr. Kent Lloyd.
Thank you for that great introduction. But I think there's something wrong. I mean, I can't have been at Davis for 28 years. I just turned 30 yesterday, so.
You have to update your LinkedIn page now.
I guess so. Gosh, what a shock. So most people get up here about to speak to an austere group and they're nervous, but I'm relieved because after hearing your introduction to this session, I'm thinking, oh my gosh, I did prepare exactly what you wanted me to prepare. How many pathologists in the room? Just raise your hand. Pathologists. Okay, thank goodness. Okay. Not because I don't like pathologists, it's that I'm sure I'll say something a pathologist will find wrong that I'm saying, so it looks like I'm covered. Well, thanks very much for coming today. So as has been mentioned, I'm from the UC Davis Mouse Biology Program. This is an outline for my talk, what I want to cover. First, just a rationale to establish and operate a mouse phenotyping core, the principles of practice for such a core, the challenges that I've faced, operations, and some things I might be able to offer you as things to watch out for. Some examples of how we organize phenotyping from both a blue sky discovery platform to more targeted, disease-focused, and then to customized approaches. And then finally, some future considerations for you. So let's go to the first one, the rationale for a phenotyping core. And I guess if I was to capture it all in one phrase, I'd say that really no one can be an expert in everything. And for this type of thing, I categorize scientists between either they're a mouse user, which is all of us, including myself, because I was trained as a horse surgeon. Can you imagine I'm doing mouse genetics? Or as a mouse biologist, meaning an expert in the mouse as a biological organism. So here's an example. So there's scientists that are doing hearing studies, learning and behavior and vision studies, and the hearing study scientist wants to use a black 6 mouse, learning behavior, they use a BALB-C mouse, and vision they've chosen C3H mice, and it could be for any number of reasons. A mouse may be cheap, somebody may have some extra mice down the hall, or that C3H mouse, even I think the coat color is just so pretty, and I just really like those. But that would be bad because these mice are deaf, dumb, and blind. So those would be wrong choices. So this is why you need to have a mouse biologist who understands the mouse as a biological organism and merely not just be a user. So there's a lot of other things that provide rationale for developing a mouse phenotyping core and operating one. Rigor and reproducibility, we've heard that now for more than a decade, and it's still very, very true that you can have more consistency, reliability in a core that's doing it on a regular basis than not. The 3 R's, of course, are very important. Egalité, or egality, of course, I put the French flag up here because of their fraternity, equality, égalité. Egality meaning that a core it doesn't depend on how much one person might have in terms of grant support versus another. A core does the same for everyone, so they treat everyone equally, and that's the egality relationship to a core. Obviously, there's an ease of availability and access to instrumentation, expertise, equipment, capabilities, procedures, capacities, and so on in a core than one would have to do in their own laboratory. And I think it facilitates and accelerates scientific discovery because just by nature, an individual will be working with other experts in their particular content areas that help to promote thinking and decisions in terms of what procedure to follow or ideas that would arise as opposed to someone working on their own. And finally, and very importantly, being in an academic institution, phenotyping cores enable training and education of the next generation of scientists. All right, what about the principles of practice? Well, I look at it as a three-legged stool, and that is any place that uses mice for biomedical research and provides core services really needs to have three things to fill out the entire content of things that scientists would want. First is the ability to handle and manipulate the mouse embryo, because there's so much we do to the embryo in terms of transgenics or knockouts to phenotyping and so on. So the ability to really manipulate, culture, maintain viable embryos for a variety of projects are important. Genome-wide mutagenesis is also important. There's even more and more need. We're just in our infancy in terms of understanding what the genome does. Isn't that crazy to think that in 2025 we're still saying that now, almost 25 years after the sequencing of the human genome, which, by the way, is only 5%, as everybody knows, of the genome. And yet there's non-coding sequence, there's epigenomic factors, there's a whole host of things that are influencing form and function. So having the ability to do that level of mutagenesis, producing mice in a reliable way that reflect those things are very important. But here we're talking about phenotyping analysis. And so the idea here is that, well, you're expert in handling an embryo, you're expert in being able to produce a model, you really need to have some expertise in a core that can now understand what this model does and how it behaves. So that's the three-legged stool. Then some other principles of practice is that by definition, a core needs to be expert in experimental design. The identity of what animal is to use, or what the genetic background of the animal to use, the power analysis, cohort size controls, and so on. The core will look at age, sex balance, genetic background, as I mentioned, diet, husbandry, and handler. All will be aspects of what a core does in terms of assessing, is this the right experimental design to move forward? I mentioned already these are three approaches, and I'll get to that a little bit later, but there's targeted platform, a discovery platform, which I like to call Blue Sky, and then a customized approach for an investigator that may have a patient specifically that has, or a group of patients that have a genetic mutation that's a variant of unknown significance. And they're asking, is this something that is actually causative, not just associated with disease? And you might think, well, why is that so important? If you have a group of patients, they're all sharing the same variant, it's still classified a variant of unknown significance, but it's pretty causative. Well, there's a real practical reason for why that's important. It's because Center for Medicaid Services, it still exists, I think, and third-party payers will only recognize treatment for patients that have likely or known pathogenic variants. So it's really important to the patient as well as to the physician. If you can help them convince the College of Medical Genetics to classify a variant as pathogenic versus a variant of unknown significance. Obviously, there's speed and efficiency. I put here clinical utility because I probably hold this naive vision that someday we'll be able to rely on mouse models and phenotyping data to contribute to a clinical examination of a patient. When you submit a CBC, you expect to get that CBC back in a couple of hours. You do another test that might require some culture and so on, it might be a few days. Well, right now, our ability to do phenotyping, as everyone knows, can take months to do, not only producing the animal, but actually phenotyping. But there are some ways to actually speed that process up. And even if we get it down to weeks as opposed to months, I think that would go a long way to making them clinically util. They're imparting upon them clinical utility, but that may still be way off. And then, of course, a big help in this is analysis interpretation. So having somebody part of the core that understands the type of data that will be that will gain from a test or a procedure, phenotyping test or a procedure, to be able to do statistical analysis, and then take on the next step and interpret that, or help the scientists to interpret that for their studies is very important, very important. All right. There's numerous challenges, of course, and the number one here is using a mouse to phenotype potential human disease. How does it translate to its translational relevance? And so I always like this paper because, well, I don't like the paper, but I use it because of the example. This Prince paper in 2011. Basically, this was a paper in which they tried to reproduce a number of studies, not all animal studies, but there are a considerable number of these, to produce studies in oncology, women's health, and cardiovascular disease. And what I want you to focus on is really this part of the pie. And only about 25% of those studies could be reproduced in a by commercial entities. So that's really important that we ensure that the models that we're producing will have a translational relevance. They may very well be, but it could be that not all the conditions of the experiment were published. Maybe only one sex was analyzed, not the other, or details were missing in the paper. Again, going back to rigor and reproducibility. Certainly other challenges, and I don't have to convince you of that, but cost and sustainability with the price of everything going up, and the need to retain expert staff, technically trained staff, who are getting costlier and costlier. That remains a problem, a challenge. The expertise, the capacity, and diversity of fields. The more we do research, the more questions we ask. The more questions we ask, the more ability to be able to address those questions. And therefore, that requires a variety of expertise, large capacity of holding rooms, phenotyping capabilities, as well as a diversity of fields. And then there's this interesting one here is new alternative methods, NAMs, which some people call non-animal models. And no less than the NIH, is investing heavily, or at least they were, in complementary or complement animal research and experimentation, looking at in vitro, in silico, and in chemical approaches that don't require animal studies. There's nothing wrong with this, except one thing, and that is if it's attempted to make patient management decisions based solely on this, unless there's absolute sufficient information to demonstrate that these findings are relevant to a living organism, then this could be a bit misguided. Totally in support. For example, some of the results from these studies perhaps will require verification in an animal study or in a living organism. I think that still needs to be kept in mind. All right, let me get to these three platforms I was discussing, I presented to you earlier. So this first example is the discovery phenotyping. Again, I like to call it blue sky phenotyping because we want to take no pre-assumptions as to what a gene or a gene mutation might do. So an example of this is the knockout mouse phenotyping project. It's funded by the National Institute of Health. It was initiated with the Common Fund. It's now moved to across the different NIH ICs that are funding it. It's the US component of the International Mouse Phenotyping Consortium, with the goal here is to produce a knockout mouse for every gene, every human orthologous gene in the mouse genome, and assess its function. When we started out with this project, only about 3,000 genes did we know the function of, 3,000. Today we know the function across the board for about 12,000 genes. So with about 20,000 genes, we're well on our way to completing this mission. The idea is to functionally annotate the dark coding genome, or those genes for which there was very, very little known. And we've done this by incorporating numerous components to this, looking at sexual dimorphism, pleiotropy, viability of the developing embryo, fertility, and aging. So it's been a pretty comprehensive study. This is an example of the pipeline, the adult pipeline. We do 8 males and 8 females between 4 and 16 weeks of age with the numerous tests, as you can see along here, covering about 10 body systems. We're missing lung, which is unfortunate except for pathology, so shout out to the pathologists there. But there's certainly some functional studies we could have done in the lung, but really wasn't amenable for high throughput phenotyping as we had here. And also these tests were done and selected, the order of these tests were done to avoid one test interfering with the results of the other test, which also is very important. Again, that level of thinking, which is important, and I think what a phenotyping core can provide than what many people would consider. And then at the end, the terminal procedures all listed here. We also have an embryo pipeline, and when we look at the time of death between 8.5 and 18.5, collecting embryos at numerous points after time matings, and then assessing the best we can through morphological assessment the cause of death, or at least the tissue of origin of death, and then making those available for further workup. We also had a small cohort that did late adult pipeline, or an aging pipeline, and then we had an essential pipeline looking at those in which these mice were, we could not produce homozygotes, and the heterozygotes were the only ones available. So a paper out of this in 2017, 360 models of 889 Mendelian disease gene associations. And what I want to point here is that 72% of these associations were novel, which is really, really good to see. There was a large percentage of those that we did which were consistent with which was already known in the literature, which is important because it verifies and validates the procedures we were doing, but 72% of these are novel. And this was 2017. We've had numerous papers since then to continue to expand on this. Okay, targeted phenotyping. Example of this is the Mouse Metabolic Phenotyping Center Live. It's funded by the NIDDK, and it's a national consortium of 4 centers and 1 coordination unit. And our focus is strictly on in vivo analysis of mouse models, and in vivo, ergo, why it's called live. And the idea here is to reveal the heterogeneous mechanisms of diabetes and obesity. And we support new investigators to expand the research workforce. So this is how the pipeline looks here at Davis. So we start here. Basically, does a PI have a mouse? Let's take this one here first. So yes, so they ship it to the animal core. The animal core does a number of procedures to verify the identity, viability, so on, of the mouse. Can go for a number of other subcores here in order to do other procedures. But then samples from these animals, or the animals themselves, can go to 2 other phenotyping cores. One, metabolism, metabolic health, to do an indirect calorimetry, looking at xenometabolism, endocrine function, glucose tolerance tests, and so on, or the physiology behavior core to look at a number of different functional aspects of different systems involved in diabetes, peripheral neuropathy, sensor gating, and exercise intolerance, and so on. And the interesting thing is that although all this part in color is funded by the NIDDK, because it's at Davis, we have then a whole host of other cores that are available that if we find something for the investigator in any of these areas, well, we can ask them, well, do you want us to send these samples maybe to the assay services core to do further hormone and metabolism analysis? And then we get their permission to do that. And then those charges goes directly to that core for those services. So that's how it works at Davis. And the other centers work very similarly. And here's an example of something more very specialized that we do. We can create bariatric models similar to the types of surgeries that are performed in humans, in this case, vertical sleeve gastrectomy. We also do Roux-en-Y's, and we get the kinds of results using on these mice from this procedure as we see in human populations. And then there's customized phenotyping. This is the program that I direct, the UC Davis Mouse Biology program, where we make unique models such as patient-specific variant models, PDXs or cell-derived allografts, or do environmental challenging, very, very specific to a patient or group of patients. We combine a diversity of scientific expertise, technical capability, resource-related research, service, and training all in one. We've been doing this since '97, and we try to allow users to use this core that best suits their needs. So either here's a mouse, do these tests, give us the results, and have a nice day, and we can do that for them. Or we can be a laboratory, an extension of their laboratory. Or we can actually be their virtual laboratory. What that means is, and I have several of these, a clinician scientists who's seeing patients all day. They don't have a lab, but they have an office, they have a computer, and we talk to them, and they basically use our laboratory as if it was their laboratory. So we do that as well. It's fee for service, and there's cost sharing as well as institutional support. So this is how access looks. Starts at the top and work our way down. UC Davis on this side, mouse biology program on this column, and this community outreach, research community on here. So basically contact us, we work with the project. They work with our project navigator. We do consultation, we develop a project plan. We then send them through send their mice or their samples through various cores, get a project file and their project results, and we try to encourage them to, and we help them to share that, either their mouse or their resources, the data, in addition to their publications with the research community. So here's an example of patient variance in mice where we work towards driving causation from an association. So this is one we published, UBE4A. This frameshift mutation is seeing 8 individuals from 4 unrelated families. They had a syndromic intellectual disability with global developmental delay. And the mouse model, which recapitulated this, showed exactly the same things plus more. And so why that plus more is important is because then sometimes these things we find in the mice, they will go back to the patients and look for them. So this has worked out quite well. We have several examples like this. And then you all remember COVID. Remember how all of a sudden we all became infectious disease experts, right? I have no idea anything about it, but I said, I think they do need better mouse models. So we made an ACE2 TMPRSS2 knock-in, and now we have an ACE2's TMPRSS2 furin, almost forgot there a second, knock-in. So humanized knock-in of all 3 human expressing genes in the mouse. And this mouse then was susceptible to SARS-CoV-2 infection. It seroconverted, established the infection, and it survived. And it all had phenotyping characteristics similar to what we see in people. Okay, and this is a new idea we have, which is actually going up for review on Friday at the NIH. Are they still having study sections? I'm just kidding. So yeah, no, that March 27th. So this will be under review on March 27th. You're all here, so I assume you won't be there. But if you are, this is really good. So basically, we're putting together a section where it's trying to, again, remember that idea I said, to move things from the doctor's office back to the patient as fast as possible. So the idea here is we've coordinated a preclinical, co-clinical section, bioinformatics, disease modeling unit, resource and services section, all organized by a coordination section in order to get nominated models to make and get them back out to the community, especially the doctor's office, as fast as possible. So future considerations, it's important to leverage the use of large language models, artificial intelligence, and machine learning. I think that's probably the suggestion for every future consideration in any talk nowadays is AI. The depth and breadth is very, very important. As we know more, we want to know more. And so having that depth and breadth is very important in your people, your expertise, your capabilities. We need to go beyond the genomic coding sequence, as I mentioned already, the non-coding sequence, the epigenomic sequence, and so on. We have to also interface, collaborate, and consolidate with other cores, animal models, organize, and even the non-animal models, or I can't remember the official name now that I said. I was so snarky about the other one. And then also, let's not put all our eggs in one basket, right? You're not surprised by that, okay. So here's an example of something we've considered and we're putting together with Dr. May Leonard, who's in the room. One contact comprehensive phenotyping solution. So the idea here is one email or one call gives an investigator access to a whole host of services, maybe under different physical roofs. In fact, these are under different physical roofs, but it saves that investigator time to try to find them all out. So we get this group of people together that are experts in all of these areas and help work out their experimental plan in order to help them proceed with their research. So again, it's a way to facilitate their activities and make it as simple as possible for them so they can get back to doing science. So we call this the Integrated Phenotyping Consortium, so we're very excited about this. All right, final thoughts, I promise, I'm almost done. There's a lot between the lines in this slide. And I made sure not to put, the insignia for the hotel and the meeting and all this because it's just my personal thoughts. I don't have to convince you there's a struggle for placement of what cores are and where they need to be, or an understanding of what they are, or the funding, right? And of course, there's new realities of the risks to direct funding for research, which is occurring as I speak and will continue to go for a while. So we can either complain about it, which I think we should, or we can, what I call, contain it, and that is find a way to respond appropriately, so making lemonade from lemons. I believe what's happening now will actually lead to an increase in demand from cores, and especially their expertise, technologies, and services, because think about it. What do cores and shared resources provide? They provide things that scientists don't have in their own lab, otherwise they do it themselves. With the way research dollars are flowing for direct research, they're going to have less and less. They're still going to want to do their science. So it's possible, actually, this could be an uptick for business for cores. I'm not hoping for that to happen for that reason, but I think this is something to keep in mind. So there's an opportunity here to revise some of our relationships, not only with the dark side, the administration, but also with PIs. Look, I'm on the dark side too, so I'm just joking. And other cores too. I've been a big fan. Here I am, I'm a mouse person. It's very funny for my basic training, but mouse person, but I've been a big fan that I should be speaking with zebrafish people and worm people and fly people and other people, right? And we just don't do it, and I don't know why, because there's so much value in this cross-species interaction and interfacing that we could do so much more together than before. So I think we really have now, let's use this opportunity to revise these relationships. I think we need to enhance operations, reduce costs, and retain and grow capabilities. And all of it is why? Why are we in this business? To advance science and technology. So I'll end there. Happy to take any questions maybe afterwards.
Thank you so much.
All right, thank you.
I think we probably have time for maybe one question in the interim, and then we'll have more opportunity at the end if anyone has a question. Dr. Lloyd? Or,
Is that clear, huh?
Is it possible, could you comment a little further on the data repositories for things like Comp2 and MMPC Live? So if you're a part of a smaller phenotyping core and you have an investigator who has a mouse model for a particular gene knockout and you're interested in what phenotypic information is available about that mouse model, or if we potentially have additional phenotypic information, is that like an interactive database, or how does that work?
So you can access the databases for both the Comp IMPC and MMPC Live. You just pick those two out, and those two I'm involved in. So it's freely available on the website for Comp or IMPC. It's impc.org. For MMPC Live, it's mmpc.org, right? And there at the top on the webpage is data access or find our data or something like that. I think the IMPC one is really superb. I think the MMPC one is good. It could be better, right? But I'm hard, the IMPC one was good before too. And after a lot of interaction and input and yelling and screaming, it got really exceptional. I think the MMPC, we're on the way there. So in order to access that data, it's freely available to all. Is that what you were asking? And then in terms of, the other part of that is, well, what if you have a mouse or you have data that you want to deposit data? So that's a really good point. And there's mixed feelings on that. On one side, some people feel like in let's say the IPC program, the IPC is set up with very standard protocols. This is how these procedures were done. It was all done in this one strain of mouse. Well, if an individual from outside doesn't follow that protocol, it's hard then to intermix that into that database. Others, though, on the other hand, feel like, well, if you don't have information on that gene, then why not include it? I think there's good points on both sides. I come down on the first, though. I think there needs to be another source, a database where someone can go to deposit that data. And there are a number of places for that. Is that what you're getting at?
Yeah.
Okay, good. Uh-oh, Denise.
Hi, Ken. So I'm interested in what you were talking about when looking at non-federally funded research. Mm-hmm. And what I wanted to ask specifically is, can you talk to us about opportunities and challenges that you've had with working with industry partners? You know, what have been your challenges? What have been your successes?
Great question. since you know me, you could have plugged me that question beforehand. Anyway, so I have to know all of a sudden. Now, how well does he think on his feet? Huh. Okay, so here's a big mistake I made in the very beginning. I thought, I'm going to make this program really big. I'm going to go grand, and I'm going to go to Pfizer. I'm going to go to Regeneron. I'm going to go all these big places. And I couldn't do it. And why? They don't need me. What I realized is they don't need me, but the little guys do. The ones that are, 5, 10, 50 people, have limited resources, absolutely don't have any animal facilities, right? They're the ones who need my services. They also need the expertise and capability and knowledge, technical abilities that I have and my group have that they don't have in their own. They're very expert, let's say, on a particular drug or a particular pathway they're looking at. So they really need my input. So in the beginning, another mistake I made was I thought, well, they must already know what they want. I'll just say, this is what we have. And it was hard to get takers until I started saying, well, wait a minute, let me actually talk to them. So it was a bit of a heavy lift and spend time to work with each one that would reach out to me and have a conversation, find out what they wanted. And after we got past the NDAs and all that, we opened up some really great opportunities there for them asking for services. The third thing is, in a way, it was a good challenge. We would have a commercial entity right now, for example, is a good example of this, who wanted technical expertise that we didn't have, but we have the foundation knowledge for how to do it. In particular was how to manipulate early, very, very early pre-implantation stage embryos. They wanted something very, very specific. And we agreed to work on this, develop it, and then provided them service. And now they won't go anywhere else. They want to stay with us. So that was also, just between you and me, that was also a good practice builder for ourselves, because now as they think to maybe reproduce that in their own laboratory or maybe elsewhere, it's going to be hard for them to do it, because now they're in the process of drug discovery or drug development, and they don't want to slow that down. They want to keep moving forward. So if we're providing them that service and being attentive, being responsive, communication with a big C, we'll keep them. And we're getting paid quite well for that. Did that answer your question? Okay.
Thank you.
All right, thanks.
Okay. Our next speaker will be Dr. Eugene Chen of the University of Michigan at Ann Arbor. Dr. Chen received his medical degree from the 3rd Military Medical University in Chengqing, China. So we did the veterinary side, now we're doing the MD side. Very equal opportunity here. He earned his PhD in molecular genetics at the Western University in Ontario and completed postdoctoral fellowships at the University of Toronto and at Brigham and Women's Hospital and Harvard University. He has been at the University of Michigan Medical School since 2005 and is currently the endowed Frederick Hutewell Professor of Cardiovascular Medicine and the Vice Chair for Basic and Translational Research in the Department of Cardiac Surgery. His research is focused on the molecular basis of obesity and diabetes-related cardiovascular diseases and on the development of new drugs and technologies for the treatment of diabetes and its cardiovascular complications. So to this end, he is the founder and director of the Center for Advanced Models for Translational Sciences and Therapeutics, or CAMTRAST. This center, established in 2012, is dedicated to the development of animal models for the rapid facilitation of bench-to-bedside translational research. Dr. Chen and his team at CAMTRAST are among the first in the world to establish rabbit embryonic stem cell lines to generate cloned and gene-targeted rabbits and to produce knockout pigs. He will be speaking to us today as the director of CAMTRAST and will be giving us some perspective on the contributions and the potential for non-murine animal models in translational research. Come on up.
Okay. Yeah.
There we go.
Okay. Thank you, Ingrid, for inviting me here. I'm going to use this opportunity as a perfect and this talk after Ken and talk. And if you want to make a model, go mouse model first. If the mouse model cannot mimic your human phenotype, and before you go monkeys and come talking to us, we might able to lower your cost, make rabbit model is able to mimic and the physiology or pathology in the human disease. I was a cardiologist. The reason I'm come to the rabbit model, I will share with you in the past 20 years or more of the frustration and what we do in translational research for the cardiovascular therapy and drug development. As, if you have any interest and know the platform at the University of Michigan, you're welcome to surf the website and send a message to me. And by the way, we struggled for 20 years for NIH funding for this platform. We still have not gained anything from NIH. The entire platform, we united everyone and have rapid interest around the country and across the world. I will share with the how we made the success together without NIH funding, but we have a grant and should be reviewed and this month, but they delayed. And we asked for NIH. Say, wow, we have so many people in this country use the rapid platform right now, and you might be thinking about to fund us for enlarge the platform to advance translational research. For rabbits, I want to give a little bit more history. Rabbits is a wonderful model for translational research in the human disease. They are back to 100 years ago. Rabbit made the contribution for the first vaccine study. That's how Louis Pasteur made the success. And also rabbits is the first be used for the animal model to develop a vaccine, as you see here. All the way went to the Nobel Prize. And rabbit also is the first to used as the IVF. And the animal model made the success for the IVF. As you can see here, they also win the Nobel Prize in the medicine and for the IVF. And rabbit is widely used and well accepted an animal model to study atherosclerosis. It's more than 100 years ago by one of the scientists in Russia started use rabbit model to do cardiovascular research, especially for the atherosclerosis. Rabbit is a model for people. When I was in training, we know it's working for study atherosclerosis. And back to the statins discovery, all statins are failed in the mouse model, the ApoE model, the LDL model. The statins is not working until the Watanabe rabbits, the Japanese model. And when you tested statin, it's working, and it's how a wonderful, great drugs and to beneficial cardiovascular patients has been identified, start from the rabbit model. So back to 20 years ago, when we look at this news, and the Congress at that time debating should NIH get any more money or yes or no, they use a very simplified outcomes. They say they only look at FDA's new drugs, new device. They say, well, you have more money doubled. In that window, we didn't see new treatments or new device to the patients. So that's the way Congress at that time used this information against the scientific community. But we think about it's the way, and the pharmaceutical also has a similar challenge. They say any discovery, many of the high leading people from the mouse model, and then they have trouble to all the way push the drug development. They say only 20% of the high impact from a couple of the big farms. So the challenge for us especially for people like me in the cardiovascular medicine, we know we need better model to do translational research. And we want to know what will be the best model for us. As we know, mouse model is wonderful for mechanism study in the cardiovascular medicine. And we want to know pigs and rabbits. We know by history, 100 years, we know it's good for translational research. But we don't have a platform to make transgenic pigs, knockout transgenic rabbits back to 20 years ago. So while the rabbits, I give you some of the examples because if you look at the heart, the human heart and the mouse heart, their anatomy are totally different. The heart in humans only beating 50 to 80 per minute. Mouse beating 500 to 800. So rabbit, it's about 120 or 180. And the mouse and the hearts, if you look at the myosin heavy chain, they are alpha myosin heavy chain, the humans, beta myosin heavy chain. So rabbits has many of the fundamental similarity to human hearts and the human cardiovascular system, but it's not the mouse and the anatomy. And we know there are many of the good reviews in the community to compare the rabbit model, mouse model, and the human cardiovascular system. And the rabbit is the animal model good enough to mimic a human cardiovascular system. So we are frustrated, but I'm not the only one. Many other leading labs in this country back to 20 years ago, like Gideon and Collins, and who was the cardiology chief at Brown. He's doing heart and arrhythmia research. And he's the one also actively generating the rapid model in the country. So if you look at what we are doing in the cardiovascular medicine, that's the #1 killer. And in US last year, more than 33% of people die in this country. It's die in the heart attack or stroke. In China last year, it's 44%. So it's the #1 killer, still the #1 disease. But if we look at the reason, they say 100 reasons might be as a risk factor, and 100 genes are contributed to genetics. If you want to simplify, maybe 50% and the risk, it's the genetics, 50% it's the genetics. So as I mentioned, when this information I just shared before, the rabbit is working in the community for cardiovascular and statin development. And then I want to bring the people together back to 20 years ago. I want to enrich and Watanabe and Watanabe rabbits. So back to 2004, I traveled to Japan, to the Watanabe group. I say, that's the problem in the community. As a cardiovascular scientist, which you are, and I'm able to partner with one of the junior faculty at that time, Dr. Fan in Japan. I say, if we united, put the effort together, call everyone around the world working on the rabbit in the cardiovascular medicine, can we start meeting each other? And we did. And we started the first meeting in 2005. Every 2 years, we bring the people in every other country have an interest in the rapid model for cardiovascular drug development. And we did in 2014, and every 2 years, and you see we're aging. And in 2024, and we bring all of the people, last year in Ann Arbor, it's more than 150 people from more than 20 countries. So they always bring everyone together. The reason when you bring all interests together, when the NIH is not funding us, might be people in Japan get money, in China, in France, which exactly the case. So when we bring the people together, we say, well, we can build models and together to, some of the examples I use in the cardiovascular medicine because I'm doing cardiovascular medicine. One of the very typical argument, if you see a cardiologist in the past 20 years, they always ask you the CRP level. If you ask how many people believe CRP, it's a good question. Does the CRP is a risk factor or just is a biomarker? So what we did, if you look at the biology side, the mouse CRP is completely different from human CRP response. As you can see here, the human CRP response, you can get 10,000-fold when anyone have an infection or have an injury, but a mouse, it's pretty much, it's no response. It's no way you can use mouse data CRP biology to do any prediction of the human biology. So we made a CRP transgenic mice back to 20 years ago. We make different dosage of the CRP, mimic the human high, low, and risk of the CRP level. A long story short, doesn't matter which level of the CRP, your overexpression has no contribution to the atherosclerosis pathology. As a conclusion, what we discovered in the rabbit model CRP is just a mark. It's not a contribute or risky and trigger the disease. Therefore, for pharmaceutical, for whatever in the practice, in the community right now, we know just a mark. It's no reason to do any drug development, target CRP for treatments. So with all of the story I shared at the University of Michigan and back to in the 2012, they say, well, you did so much. We want to build a center for this rapid platform. That's how we'll get a big enhancement from institution and start able to build the core. When you build a center, you not only need animal and a facility, you need stem cell biology experts. To make a long story, we're able to recruit the 6 faculties in the different domain of the excellence and build a center together. So that's how we did in the Michigan. And another thing I say, when NIH is not give us the funding to the rapid journal, we have to unite the internationally together. I'm the PI leading that effort. We're able to pull the money together from the different countries. We are able to provide the entire rapid sequence to the scientific community. That's how we published, and that's the international efforts. Right now, if you make any knockout genes in rabbits, you have the access to the. So, anything in the beginning is not easy. Before, right now, 20 years ago, it's very challenging to make knockout on rabbits. And what we are doing at that time, we do make knockout fibroblast. And then we try to clone it to make rabbit models. So we have to do 100 times of the screen, and then you get 1 positive. And then you have to do 100 times micro-injection, you could get a knockout rabbits. The answer is yes, you can produce knockout rabbits, but it's meaningless because you cannot help anyone by that cost of the effort. So the bottleneck at that time, once we didn't have yeast cells in the rabbits, and the second, the efficiency for cloning is too low. And we all know by 2020 the game changed. So the gene editing wins the Nobel Prize, and it's a Cas9 technology and moving very fast. We are very beginning, we're actively in the field and doing gene editing. We are also among the first group and publish the gene editing Cas9 technology to generations of rapid model. We generated more than 10 of the rabbit model in the beginning with the Cas9 technology. We published in the 2014. That time, all the model at that time, it's the cardiovascular model, because I'm working on the cardiovascular medicine. And then when we build the models, we have to make sure we have efficiency improve, and the editing platform, we made progress. And also we have a Michigan Cas9. We call Michigan Cas9 so far, it's the high efficiency for high fragments and recombination, and with the lowest and off target on the market at this moment. And it's a Michigan Cas9. We're more than happy to share if you have interest. And the way when we build so many cardiovascular medicine and models shared in the community, people start approaching us from all domains of the science, which I have no idea before. For example, the leading HIV research, and from Germany, from this country, the scientists approached me and told me, only monkey, rabbits, humans, 3 species can be infected by HIV. So I have no idea. And then we generated a whole bunch of the rabbit model for people doing HIV research. And then the same thing, the Johns Hopkins and Columbia and Michigan, the Eye Institute, the people and teach me the rabbit eyes is good enough to mimic a human eye disease. They ask us build many models for more than 5 leading institutions in this country for eye disease. This group of people, they are very united, the eye doctors. They are united together and talking to FDA. Last year, they're able to convince FDA accepted if a treatment for eye disease in the rabbit model is working, it's good enough moving to human clinical trials. So that's only for eye disease. So when people united and put science together to argue, you can change the policy. So we have another popular model we built. It's the email deficiency and the rabbits. Right now, there are more than 10 other institutions use the email deficiency rabbits for all kinds of disease. We are happy to share with the scientific community. And eventually, what we are doing in the Michigan, we want to build rabbit model and mimic the human disease, and then for gene editing and therapy. That's what is the program. And for myself, it's focused on cardiovascular medicine and gene therapy. And we hopefully for one shot for cure. And what I did in Michigan for my own research program, I have a human genetics program where I identified and many of the genes and disease and related genes as I list here in the past 10 years. We build a rabbit model. We hopefully do one shots for cure some of the genetic disease. And if you look at a database, there are enough genes always there. 99% of the genetics gene and mutations has no treatments. If you pay attention to single gene mutation to the single one phenotype, that's more than 3,000 diseases always there. That group of the disease could be treated in a 5 years window. It's my prediction. Many of them, when the gene editing and it's working and an animal model is ready, and when you treat the animal model, it's working, could be a human and treatments for this rare disease. So that's how we did 20 years in the Michigan medicine. We built a large and rapid platform over there, help the community. If your mouse model cannot mimic your human disease, and think about a rapid model might be an alternative. And then we want develop a Michigan Cas9 for gene editing and delivery. Hopefully, will bring a new class of the treatments to benefit and patients. I'm focused on the cardiovascular medicine. As I said, the people in other community doing the genetic disease treatment, we come more than happy to help. As I say, ChemTrust is not by myself. We have many of the faculties always there and working together with different expertise. That's a group of the people, as you can see here. We have many of the fellows. I'm working for all and the faculties to help this one. We also have four of individuals. It's very important. There's a strong argument for NIH cannot cut indirectly cost. So because the people help us to manage the center for finance, for administration, for grant submission, that's all heavily rely on indirect cost. If we didn't have this 4 individuals, the center is not going to function. So they always take an army of the scientists, a village of scientists for every country, every institution has a common interest to working together to achieve the success. So I started more than 25 years ago. I want to mention one of my friends, Jerry Young, passed away when he was 49 years old. He's the first individual cloning the animal in this country, the call, the MB call, and 2 of us getting together back to 25 years ago. I say, I need your expertise to help me make rabbit model. That's how I'm able to make cloning a rabbit model in the beginning. So, thank you. I'm stop here. Thank everyone for your attention.
Go ahead.
Jennifer Koblinski from Virginia Commonwealth University in Richmond, Virginia. I direct the Cancer Mouse Models Core, and we do obviously some survivorship studies and doing some work related to doxorubicin and its effect on the heart, right? So seeing what you're talking about here, I don't even know how many tumor models there are in the rabbit right now. I only work with mice. But do you feel like maybe some of these studies should be being done in rabbit models? And how many of them are there some good tumor models, like breast cancer models, in rabbits? Do you know?
We didn't have that many cancer people use the rabbit model at this moment. I can only comment what we are doing for atherosclerosis, for the heart attack, for the stroke. And if you're doing the translational research, rabbit is the model. Mouse can help anyone understanding the mechanism, but it's not going to bring a translational to treat an atherosclerosis.
Okay. Yeah, normally how many rabbits do you use when you're doing these studies? Like what is the variability between the rabbits? Like in mice, we might be using like 12 or 15 for each group.
So we use N equal to 10 to 12 for each group.
Okay.
Yeah.
Yeah, but do you think that, I know you're talking about atherosclerosis, but do you think that the damage of the drugs, are similar in the mouse to the human, or do you think they would be better to do that in a rabbit model?
I tell you, wild type mice, no mice is going to die of the cardiovascular disease. They never have atherosclerosis. They are well protected because they are HDL mammals.
Okay.
Humans, we are LDL mammals.
Okay, thank you.
Yeah.
Thanks. Do we have any other questions for Dr. Chen? Oh.
Hi, I was curious, do you have experience with using your transgenic models with atherosclerosis with device development? Is that a common thing?
The answer is yes. And we published in Nature bioengineer imaging a couple years ago, and working with the bioengineer people, try to develop next generation of the imaging. Yeah.
As far as like stents or drug-eluting stents, have you worked with any of those?
I'm not working with them directly, but in the Michigan cardiology, yes, they actively use rapid model for STEM development. So if you have interest, I can connect you to people when you're home.
I worked with a pig CRO years ago that was working on device development in the pigs, and it's curious to hear all the work you're doing in the cardiovascular model in the rabbit compared to what may have been also done in pigs. Do you favor one or the other with the devices?
So the device development different. For my understanding, the device company, they more prefer use pigs instead of rabbits. But for people doing drug development in the cardiovascular medicine, because we approached by, I have not mentioned, all of the major farms, they used my co-facility in the past 10 years, develop a rapid model for the drug development. That information I cannot share.
I have a question for you. One of the main challenges, I think, for people using non-murine animal models, if you're trying to characterize your model more fully, is reagent availability. So like immunohistochemistry obviously is geared more towards human samples or to mouse samples. The rabbit contributes there by helping us make the antibodies. But for your models, I know getting sufficient validated antibodies must be a challenge. Are you using other techniques like ISH or spatial biology for characterization?
That's very good questions. So because many of the research antibodies are generated by rabbits.
Yeah.
And therefore, when you're doing the rabbit biology, you are in trouble.
Yeah.
So what we are doing in this community, we're generating the antibodies. In this scientific community, we share the information, we help each other. So that's how we are moving forward. And with more and more people use the rapid platform, the situation and getting improved every year. Yeah.
Is that information available, like as far as antibodies that you've validated or have?
Nobody put a database together, but because the community is still small.
Yeah.
Yeah. It's no more than 100 labs in the world.
Thanks for bringing that up. Actually, we're going to be talking tomorrow about is it tomorrow? It's Wednesday.
Wednesday.
Okay, Wednesday, we're going to be talking about describing an antibody database that's being put together by members of ABRF right now. And so we're looking at things like this, getting data on, specificity towards different organisms. And so, having specificity towards a rabbit is a perfect example there, where perhaps we could help and collate some of that data together for people's use. So
Thank
a little
you.
plug, that's all.
Thank you. So by the way, I'm going to emphasize the mouse model still the best moving forward for your scientific and hypothesis. And although I'm running the rapid platform, my lab has average 800 to 1,000 kgs of mice every day.
Thank you. That was a great, excellent talk. At this point, we have about a little less than 10 minutes left. I'm just going to open it up. If anyone has any comments, either from their experience in their own core facilities or questions for either of our speakers, I'll just kind of jump down in case there's a question for Dr. Lloyd, if you want to hang out here for one more second, Eugene. But does anyone have any comments or questions or experiences from their own? Does anyone here, I guess show of hands, if you run either a phenotyping core or a histopathology core or work with a diversity of animal models, hopefully. So I guess for myself, one of the things I'm hoping, I think, we all have our various communities, either through our research focus or for myself, through my field, through pathology, where we communicate to other folks running similar cores. I think for many of us, at least for me, I've had to learn sort of the mechanics of running a core and the administrative headaches and the sort of service aspects all on my own by painful experience. So I think as through communities like ABRF, hopefully we can kind of build a little bit of an animal niche to try to get some of those questions in the context of animal-related cores. I don't know, just as Eugene said, no one can do it by themselves. It takes a large community. So hopefully we can sort of facilitate some animal core-related discussions going forward as part of ABRF and bring some of these resources to bear. Does anyone have any questions or comments?
I think we talked about this last time, and right, with Amanda, you guys are unicorns, right? And I think that's what, there's not a lot, unless I'm wrong, but from
Denise
what I
and Ken are
understand-
also unicorns.
Right, there's not that many vet pathologists, and I think that part, I somehow fell off that histology group, I gotta get on there. But I think that's another part, like helping make that community, because Charlene, she's not, there you are, because Charlene was telling me they have 4 there, but like their connections at North Carolina State, right? Or, right, is that right? North Carolina State with the vet school, you were saying? That's part of it, but I think that's really important what you guys do, and we did find someone at UVA, right? But VCU, we don't have anyone. And I know when I was at Northwestern, there was no one else either there, right? So having you, you know, being able to, and Gordon told me if I tried to steal you, and I was like, I know she won't come. Right, but it's not that, it's just, you know, that's the other part, creating that network of when you guys can have time and who can have time to send in, who's, maybe even what are your expertise in different models, like having that listed so that we could send those like we just talked about in the cancer center thing, right? Yeah. That consortium.
Yeah, I mean, we'd love to chat. There's actually, veterinary pathology is an extremely small field, and everybody either knows everybody else or you're like 1 degree removed. But there is a comparative pathologists consortium, and that's the very small niche area of veterinary pathology that works in academic cores or with animal models of human disease. So there's sort of the diagnostic arm from the vet schools, and there's the folks who work in pharma who have a lot of mouse expertise, but their attention is sort of elsewhere. But there is a comparative pathologist consortium with some very experienced people that, you know, we'd be happy to kind of facilitate discussions with. Sure.
So great to see these topics coming here at the BRF. Some of us veterinarians, pathologists are all getting more involved. I had a question about your presentation. You know, the hospital, the mouse hospital concept, I think, something that you showed in that as a proposal, something very familiar in the cancer field with the PDX models. People have been trying to, you know, at some point many years ago, there was this great excitement that, you know, we could probably figure this out with PDX models with every patient, individual cancer, you know, individualized cancer therapy. It didn't go that way, mainly because it takes so much time, and the outcome, of course, is so heterogeneous. But even with all that difficulty, it is all we have to have is NSG mouse. Now, I'm wondering, when you talk about creating a transgenic mouse and the whole cycle, how long does it take? Or is it really, from that perspective of, you know, kind of answering a few patients, you're trying to create a transgenic mouse? Are there examples of how long, how complex
Okay.
is that process?
Just kind of general.
Yes.
It all depends on the approach that you take and the mutation, let's say, that you're trying to recreate. If you're merely trying to, let's say, create a SNP mutation, now we have CRISPR. You can actually make the mutation if you have everything available to you, mouse embryos and all that. You can make the mutation to have a mouse on the ground in 3 weeks. Now, that's from injection to birth, right? Because it only takes in the morning to inject, you do a transfer, you have birth after you do the embryo transfer, then you have birth in 3 weeks. And then it takes a little while, you know, for the animal to grow up, maybe to see the phenotype you have. So we're on, you know, the CRISPR has really transformed our ability to make genetic mutations in animals. So we're talking now, instead of a year or more when you were using embryonic stem cells, down to only weeks to months. So it's very, very fast. On the other hand, there are other mutations. Let's say you want to humanize an entire coding sequence. CRISPR may not work. Then you end up going back to either maybe try AAVs, or you have to do embryonic stem cells. So then it can extend that time. In general, though, if you look over history of the last 20 or 30 years, when transgenics were just getting off the ground, there's been tremendous progress and advancement in technology, which has not only improved the outcomes, but also dramatically shortened the time to get the product that you're after. And I can only envision that will continue with, of course, the appropriate support.
Thank you.
Welcome.
And can I repeat what Jennifer asked? Maybe.
Sure.
I was not sure whether you were answering that. For cardiotoxicity models, is rabbit a better model than mice? Like chemotherapy, cardiotoxicity is a big issue.
I didn't try. I'm more than happy to help you people, anyone within you want to try the rabbits for that direction. But we met many of the models for cardiologists in this country doing the arrhythmia research. So the leading arrhythmia research group in this country, majority of them right now use rabbits for transgenic study for the gene and to mimic a human phenotype. So that community I know at this moment.
Thank you.
I guess I can just briefly say, as a person with background in toxicology, I think that might also be, thank you, drug dependent, because depending on the mechanism, I don't know specifically for doxorubicin, but it might be a little bit more on the drug side. But I agree with Eugene that physiologically, I think the rabbit is very much a more translationally relevant model in terms of their cardiovascular physiology. And we've run into that in my institution. I work with a number of investigators with genetic or drug-driven aneurysm models. And the mouse, I hate to say it, I work with mouse all the time. I think the mouse is kind of a terrible model for aneurysms because they're either, if they're gonna go, they're just gonna die. And then they don't have the layers in the aorta that the rabbit does and that the human does, and the pathogenesis of aneurysm in a human is much more like the rabbit model, in my opinion, than in the mouse. So it might be a bit model dependent.
There was a study recently done in our place. In China, aclarubicin alternate to doxorubicin, they're used in human with less cardiotoxicity, and the mouse model seemed to replicate that very well.
So it might be, like I said, drug dependent in toxicology because you have that other aspect to it.
Can follow up on
Sure.
this. If you're interested in functional changes and not just dying from cardiovascular disease based on toxicity, then rats are a better model for MRI doing functional cardiac imaging. So you can switch to something in between, which is a rat. Yeah, well, speed. The mouse heart beats too fast. We can image it with MRI, but the functional cardiac imaging is not as good in mice compared to rats.
Okay. All right, I think we're getting close to our time, but I'd like to thank first of all our speakers for a really informative educational session. So thank you very much to our speakers. And thank you to all of you for coming to the session. I was a little bit worried that it was just going to be me and Ken Kim, but thank you for coming, and please feel free to follow up either on the community forum for ABRF or by contacting us individually for any follow-up discussions.